Hello again, everyone. So we will have a very short session of 2 hour presentation. I'll remind the format. It's two presentation of around 10 minutes to allow a few questions after that. So I'll begin with Christianah Omotoso from the University of Western Cape of South Africa.
Christianah Omotoso. I'm a Master of Pharmacy student at the School of Pharmacy, University of the Western Cape, under the supervision of Dr. Samuel Geya and Professor Helen Christopher. I will be presenting on the homology modeling and structural comparison of three-dimensional protein structures of selected variants of CYP3A4. This is the outline of my presentation. CYP3A4 is the most clinical abundant drug metabolizing enzyme as shown in the picture here. Its structure uniquely differs from other cytochrome P450 enzymes with its truncated G and F helices and the unique phenylalanine cluster forming a part of the active site. CYP3A4 has been reported to be highly polymorphic. This causes inter-individual genetic variation in CYP3A4 that influence the efficacy and toxicity of numerous drugs and prodrugs. The individuals carrying different CYP3A4 allelic variants have been classified as poor metabolizers, intermediate metabolizers, extensive metabolizers, and ultra-rapid metabolizer according to the metabolic nature of the drugs and prodrugs. Problem statement and significance of the study. While several studies have shown the effect of genetic polymorphism on the functional catalytic activity of CYP3A4, there is a paucity of information on the effect of single nucleotide polymorphism on the enzyme molecular structure. In this research, 4 CYP3A4 allelic variants were selected based on the report of single nucleotide polymorphism on the functional assessment of CYP3A4 allelic variants in lidocaine metabolism. 22 allelic variants were subjected to the in vitro study. However, the 4 chosen variants are those that show significant difference in the clearance of lidocaine relative to the wild type. The significance of the study is to investigate the structural differences of the model protein structure of the CYP3A4 selected variants. The research question: what are the effects of single nucleotide polymorphism on the three-dimensional protein structure and physiochemical protein characteristics of selected variants of CYP3A4 relative to the wild type? This study aims to generate the three-dimensional protein models of selected CYP3A4 variants via homology modeling, with a further study to investigate the effect of single nucleotide polymorphism on the three-dimensional protein structures. The research objective involved the collection of data and collation of the selected variant sequences of CYP3A4, modeling of the three-dimensional protein structure of selected CYP3A4, validation of the quality of the modeled three-dimensional protein structures, and structural comparison of the differences in the physiochemical and static properties of the modeled protein structures of the selected variants. For the collation of the CYP3A4 wild type and the selected variant data, the CYP3A4 wild type amino acid sequence was retrieved from the NCBI database in the FASTA format. The selected alleles of CYP3A4 for this study were retrieved from SNPedia and on the human cytochrome P450 alanine nomenclature website to generate the variant sequences with the known position of the amino acid substitution as obtained from the databases. The sequences of the selected variants were generated on Python with its underlying ByPython package, which was installed into the Anaconda working shell. The substitution of the amino acid was implemented at each predicted position of mutation of each variant as indicated in the database for the different variant sequences. To model the three-dimensional protein structures of selected CYP3A4 variants, homology modeling was carried out on Swiss model and automated protein homology server. A template search was done using the imputed sequence of the wild type as a query to find the protein structure that is most related to it evolutionarily. This was done by the blast method with the GMQ of 0.83, QSQE of 0.52, and an identity of 100%. The models of the Y-type and the 4 selected variants were built with the template. Evaluation and validation of built model was done on Safe Server, QMIS Server, Verify 3D, and the Ramachandra plot of the built model were plotted for the validation. Structural comparison of the selected C3A4 with the Y-type was done on Chimera. The prediction of the protein stability of the model protein structure of mutant was done on the high mutant 2. Primary amino acid sequence analysis of the C3A4 selected mutants were done on ExpaSI Proparam 2. The table shows the selected variant with an indication of the substituted amino acid and the position of mutation in the selected variant for this study. This is the model protein structures of the selected variants showing the secondary structural analysis, helices in red, strands in purple, coils in blue, and this is the hydrophobic surface view of the modeled protein structures. This is the result obtained from the validation of model structure, in which more than 90% of the residue of all model structures in the favored region in the Ramachandra plot, which is the characteristics of a good quality model. This is also supported by the Q-mean Z-score around 0, which shows adequate correlation in comparison of the model structure with the experimental structure of similar size. This validates the quality of the model protein structure of the selected C3A4 variant in the study. This is the result obtained from the structural comparative analysis, in which the protein structure of each mutant was superimposed on the protein structure of the Y-type. Differences in structural features such as alpha helices, beta helices, and coil regions were visualized. However, there was no noticeable difference in the features. The root mean square deviation, which is a difference in Armstrong of measurement of backbone atom, was done for each of the variants superimposed on the Y-type, and these are recorded in the table below with variant 2 and variant 24 showing the highest deviation. This is the result obtained from the analysis of all the physiochemical properties. All mutant study has the same isoelectric point of 8.27 except for variant 23 with an isoelectric point of 8.0. This is an indication of a mildly alkaline protein. The extinction coefficient of all the selected variants or mutants are the same. The instability index for all the selected mutants are above 40. With values higher than 40, there is an indication of instability, with more or higher instability in CYP2 and CYP24, which is similar to the report obtained in the RMSD. The aliphatic index, which is the relative volume of protein which the aliphatic side chains occupy, is the same and is high in all the studied mutants and the wild type, which indicates a state of thermostability. The graph, which is the grand average of hydroplasticity, is the sum of all the hydroplasticity value, is low, and this indicates that the interaction was more in between water and the protein. The graphic for the selected mutant differs from that of the wild type. This is also seen with the molecular weight, this is because of the substituted amino acid in the selected variant. There is a decrease in the stability of all the selected and model protein structure of the variant, and this is in line with the report obtained from the RMSD and the instability index from the physiochemical characteristics of the selected variant. In conclusion, the protein structures of the selected variants were modeled via homology modeling, and the quality of the model structures were validated to be accurate, as confirmed by the quality assessment tool. There is no obvious difference in the structural features of the mutants in relative to the wild type. There is difference in the backbone atom of all mutants in relative to the wild type, with the highest deviation in mutant 2 and mutant 24 as indicated in the root mean square deviation. There are observable differences in some of the physiological properties of the study mutant, specifically the graphy and the instability index. The stability of all modeled protein structures is predicted to have decreased. Future presentation would highlight the observed correlation between the structural differences and the reported functional differences. My references. Thank you.
Thank you, Christiana. So it was very interesting. Is there any question? Don't be shy in the audience. We'll take question. Okay. So the first question from David is it possible to model the effects of haplotype combination of this variant on CYP3A4? It asked model a mutant with more than one variation.
Sorry, can you hear me, please? Sorry, can you hear me?
Yeah, I hear you.
Thank you, David, for your question. Yeah, it's possible because presently what we did was to model based on most similar octane structure from the homologs that were present on the server, and with that, it's possible to also model other likable variants that are discovered with time.
Okay, thank you. Thank you very much. Do we have time for another one, maybe? Okay. There isn't another question, so we will move on. My bad. Okay. Wait a bit. We have another question for you. Why were there no significant differences?
Okay, for the observable features, there was no significant difference, but for other things like the RMSD and the physiological characterization, the stability, there are differences. Actually, this study has more to do with further characterization, such as solubility, ionization energy, sorption energy, and we're also looking at the dynamic properties of the variants, which is not a part of this presentation, but further study or characterization that we've done had actually shows significant differences. And also from this presentation is just the physical features that are not showing significant differences, but physiological characteristics, as I've mentioned, and the stability compared to that of the white type has also.
Oh, seems we have a connection issue. We might not have the end of your answer, Christiana.
Sorry, can you hear me now, please?
Yeah, I hear you now.
Okay, so I was trying to explain that it's just the physical features that we weren't able to notice significant differences, but other properties like the carbon atom, the backbone atom, and the physiological characteristics of the model structures, then the stability, we observed significant differences. And also, we are also into further characterization, which is beyond this presentation, like the solvation energy, ionization energy. We're also looking into the binding energy and the dynamic properties of the model structure. That took us into molecular dynamics, which is not really part of this presentation. So there are other significant differences have been obtained from the characterization, and that is why I concluded that further opportunity to present would involve that. Thank you. I hope
We
I've been
thank
able to answer your
we thank you.
question.
We thank you
Yeah,
for
thank
your
you.
presentation.